Hello, and welcome back to Introduction to Genetics and Evolution. This is one of my favorite parts of the course. This is what people typically think of when they think of basic genetics, and that is basic single gene inheritance. In the last video that you saw, we talked about the process of mitosis, which starts with a diploid cell having two copies of all genetic information and forming two new diploid cells that are genetically identical to each other and genetically identical to the parent cell from which they came. Now, meiosis, which is what we'll be focusing on here, you start with a diploid cell again, having two copies of genetic information, and you make a haploid gamete. It has only half the genetic information. Now, this haploid gamete comes together with another haploid gamete. Boom! Fertilization happens, and you have a new diploid cell that is different from either of the two parents. That is really cool. So let's talk about that briefly. Meiosis and fertilization are what are needed for what's referred to as Mendelian inheritance. Gregor Mendel was a monk who actually described this process very elegantly and identified how this actually worked. He lived from 1822 to 1884 in Austria. What he worked with were pea plants. He identified a number of traits that he was studying with these. So as you can see here, seed form being round versus wrinkled, seed color being yellow versus green, pod form inflated versus restricted, etc., etc. There are a lot of traits he looked at. What he found is that some forms he referred to as, quote, dominant. Dominant. The dominant meant that if you bring together a round form and a wrinkled form and cross them together, the offspring would actually just be round. It is not intermediate, but would actually just be round. So from this, he identified some simple rules of inheritance. Now we can understand dominance and recessivity from these simple forms. So let's use, in this example, true breeding green pods and true breeding yellow pods and cross them together. Okay, now when I say true breeding, I mean all their offspring are when they're bred with others that look like them, look exactly the same. Now, what we'll do in this case is we'll identify the genetic factors that they have, or the alleles that they have, using a letter. Typically, but not always, the capital letter is associated with the dominant form. So in this case, the true breeding green pods are dominant. So it is big Y, big Y. Again, it has one copy that came from its dad, one copy that came from its mom. What gametes can it give? Well, it's only got big Y, so its gamete would have to be big Y with respect to the form of this pod, with respect to it being green. So these guys right here, the yellow pods, they are what's referred to as recessive, and I'll show you why that is in just a second. They are little y. Again, they have, little y that came from dad, little y that came from mom. All they have to give are little y's. The F1s, or the offspring of these two things, the F1 is an offspring from a cross. Well, they'll get the big Y from this person, or from this plant, and the little y from this plant. So they are big Y, little y. Nonetheless, they are all green. So in this case, we have a masking of the yellow color by the green copy, right? The masking of the yellow color by the green copy. So big Y, the green one, is definitely dominant. Little y is definitely recessive because you don't see it when you have one of each in there. And what will this plant over here give off? Well, he has two different gene copies. He has a big Y and a little y. So he could give either of these to his kids. So what happens when we breed these F1s? So this big Y, little y are referred to as heterozygous. Heterozygous meaning having two different alleles at a particular gene that we're looking at, in this case being the pod color gene. Right? So they have both, they're heterozygous. So we can use what's referred to as a Punnett square to follow patterns of inheritance. Now, it's equally likely for it to give one copy as the other copy. So we'll take a boy that's big Y, little y. So here's your boy, big Y, little y. Here's your girl, big Y, little y. Now, half of the gametes from the boy will be big Y. Half the gametes from that same boy will be little y. Half the gametes from the girl will be big Y. Half the gametes will be little y from the girl. So the possible combinations, the boy might give this big Y, the girl might give this big Y, and you may get a big Y individual. Well, we know that is green. The boy may give big Y, and the girl may give little y. Well, we know when you're heterozygous, you're still green. The boy may give little y, and the girl may give big Y, still green. And only in this case, when the boy gives little y and the girl gives little y, do we get a yellow pod plant from them? So the ratio we should get is 3 green to 1 yellow. And that, in fact, is actually the ratio that Mendel saw. So here's some actual numbers from Mendel. He saw 428 green and 152 yellow peas from this cross. That's very similar to the expected numbers. So that's great. Now, Mendel put out various laws, and these laws have postulates within them. So let's go over that briefly. So Mendel's first law has 3 postulates. First, that we have unit factors in pairs. So that basically every gene, you have 2 copies of it. This is assuming you are a diploid. So you get 1 allele from your mom, and you get 1 allele from your dad. Similarly, if you are a boy like me, you will give 1 allele to your kid. Your spouse will give 1 allele to the same kid as well. There's also this feature of dominance or recessivity. You don't always see this, and it is possible sometimes to have, say, a red plant and a white plant come together and make a pink plant. But sometimes you do see this where one form completely masks how the other form may look. So you can have a green and a yellow pod as you saw, and the offspring looks just as green as the dad. And finally, we have the equal segregation of gametes. Right? So that you have these 2 factors, the big Y and little y, and you're equally likely to transmit either the big Y or the little y to one of your kids. You could transmit the big Y sometimes, a little y sometimes. You don't have any control on it, but it's a 50-50 shot. So let me give you a problem to try. First, I'll do one with you, and then I'll give you one just to try on your own. Let's imagine you're a farmer working on corn. Okay? And let's say you have pure breeding tall and short strains of corn, and you've heard that this is caused by a single gene. This, by the way, is very, very unlikely, but let's just pretend it's. So you cross the tall and short strains together, and in this case you get strains that are intermediate in height. So in this example, we don't actually have dominance like in the previous example. So you cross these intermediate height corns together, what would you see? Well, let's work through this together. So again, we started with pure breeding tall and short strains, right? Let's imagine the alleles we're working with are big T for tall and little t for short. Okay? So let's imagine these are the 2 forms we're working with. Now, what would these intermediate strains be? Well, since we said that we're crossing a pure breeding tall, which presumably was big T, to a pure breeding short, which presumably would be little t, the intermediate ones would presumably be big T, little t, big T, little t. So what are we going to get in the offspring? Well, there's a lot of possibilities. We could get big T, so this is big T coming from dad, big T coming from mom. We could get big T, little t. We could get big T, little t, or we could get little t, little t. So in this case, this is little t from dad, big T from mom, or in this case, this is little t from dad, little t from mom, etc. So what do we see with this? Well, in this case, we have 1/4 here would be big T, which we've identified that as being tall. Big T, little t, we've identified that as being intermediate, so it'd be 1/4 intermediate. Okay. Down here we also have big T, little t, so 1/4 intermediate. And then finally, 1/4 short. So basically a quarter would be tall, half would be intermediate, and a quarter would be short. So the ratio would be 1 to 2 to 1. Okay? Let me give you an example now to try yourselves. So this is starting similarly. You're a farmer working on corn. You're pure breeding tall and short strains together. You cross the tall and short strains together and you get intermediate height corn. This is again very similar to the previous problem. You cross the intermediate height corn to the tall corn now. So you're crossing intermediate height corn to pure breeding tall corn. What will you find? Welcome back. I hope that was not a particularly difficult problem, but let's work through it nonetheless. Okay? So you started off, in this case, you had your intermediate corn. So this is just like the previous example. However, rather than breeding the 2 intermediates together, you're breeding intermediate corn with pure breeding tall corn. So this is a little bit different than last time. So here's your pure breeding tall corn. We'll just say that's the female. It doesn't really matter which one's which. And we'll say your intermediate was the male down here. Well, in this case, there's actually fewer possible offspring. So you can get big T from here, big T from here, and you get big T, big T. Here you get big T from here, big T from there, big T again. So you can see half the individuals are big T, big T. You can have big T from mom and little t from dad. And again, big T from mom and little t from dad. So in this case, unlike in the previous example, you would get 1/2 tall corn, and you would get 1/2 or 2/4 intermediate sized corn. You would not, in this case, see any short corn coming out. Now, I hope this illustrates something to you, especially contrasting to the P example. And what I hope this illustrates is that dominance does not matter in how offspring will, dominance matters in how offspring will look, but it does not matter in how gametes will pair. You know, we can use the same kind of Punnett square, for things which exhibit dominance as things which do not exhibit dominance. Because dominance only affects how things look. It does not affect how pairing happens. Okay? So let's try out a medical example in this case. So about 12% of women on average get breast cancer. And there are known mutations in the FGFR2 gene that are associated with an increased risk of breast cancer. Okay? So let's call in this case the non-mutant form big F, and let's say that they have the default 12% risk of cancer. If you're heterozygous, if you're big F, little f, you have a 20% higher chance. So you have about a 15% risk of getting cancer. If you're little f, this is the worst one, you have a 19% risk of getting cancer. Okay? Now, in this fictitious example, let's say that you're going out someplace, you're an attractive female, you meet somebody, sitting in a cafe, you have a nice chat with them, and let's pretend this is in the future. Let's say it's 10 years from now. They leave. You pull out your little iPhone, and you go, boop, and you use the DNA scanner on their coffee cup, and you say, oh my goodness, he is heterozygous for the FGFR mutation. So let's assume that you are homozygous big F, so you have the lowest probability of breast cancer, but your potential hubby is a heterozygote. He is big F, little f. And again, I put here all the probabilities so you can see what everything was. What is the probability that your daughters would get breast cancer if you were to have kids with this potential hubby? Basically, how much does it increase it relative to if you had kids with somebody who was just like you? Well, we can do this very easily. So your potential hubby, we say, is big F, little f. You are big F. Okay? So we're just looking at your daughters. We're not worrying at all about your sons in this case. Well, half your daughters will be big F. So half your daughters will be just the same as you. Half your daughters will have this mutation. They'll have the thing coming from your new hubby. So there's a slight increase, but these ones have a 12% chance of getting breast cancer based on this gene alone. Of course, we're not considering everything else like lifestyle and other genes. These ones have a 15% chance of getting breast cancer. So on average, instead of having a 12% chance of getting breast cancer, your daughters would have about a 13.5% chance of getting breast cancer. That's averaging all four of those numbers together. So really, the guy is only increasing your odds of having daughters with breast cancer by about 1.5%. So you can decide if that's important enough or not in terms of whether you want to keep this hubby. And we'll see if this sort of iPhone app ever comes about in the future. That's just a fictitious example and a humorous one just to give you an idea of what's happening. Now, we can do the same type of cross with unknowns and actually infer the parents. So albinism, being an albino, is inherited as a recessive in humans. Now, what if you have a case of a non-albino mom and an albino dad? They have 8 kids. Let's say 4 of these 8 kids are albino. Can you infer just from this information on this slide what the genotypes are of the parents? Welcome back. I hope that one wasn't too difficult for you, but let's go ahead and work this one out. So we have a non-albino mom and an albino dad. They have 8 kids, 4 of which are albino. I said albinism is inherited as a recessive in humans. Because it's recessive, since the dad is albino, the dad necessarily has to be homozygous for the recessive. So let's call it A. So dad has to be little a. Okay? Now mom is non-albino. So she could be big A, or big A, little a, right? Now, what if mom was big A? If mom was big A, and dad was little a, all the kids would be big A, little a. So none of them would actually be albino. So given that we know that 4 of the kids are albino, we can cross this out, and we can say, in fact, mom must be big A, little a. So in this case, from looking at just the phenotype, from looking at how the individuals look, that's what I mean by phenotype, and from looking at what happens with the kids, we can actually infer the genotype. Basically, what is the underlying genetic components? What alleles do they have within them, and what combination? So, I hope this was useful, and I hope you'll enjoy it. We'll do some more complicated things later on, and you'll have some practice problems to work with as well. Thank you.